So again, good morning. So today I'll be discussing the part where we have stopped yesterday. In the sense, I have given the preamble right as a day one session, sir. So our speakers have started with the network pharmacology. Some have started with the what we call homology modeling, protein preparation, pharmacophore modeling. So now I'll be discussing in depth about the QSAR modeling, how to utilize do the QSAR modeling using AI-based drug discovery. So to start with the session, I just want to give us some preamble. What is AI and what are the machine learning models available? And based on that, we'll be moving on, and I'll be showing a demo how to perform those things and all. So this is my disclaimer slides as usual. All rights one goes to their rightful owners. So first, what we should know what is artificial intelligence. Is this a new topic? No, it's a very old one. A is in the field of computer science. It's a field of computer science where with the concept of machine thinking like humans. We humans, how we think, we have to train the system, we have to train the computers. That is called as a machine, what do we call AI. So to perform tasks such as learning, problem solving, planning, reasoning, identifying patterns. In simple example, how you are using a chat GPT. Many of them people are using chat GPT, right? If some question is asked, just throw to the chat GPT, it will be giving some answers. So in the sense, it have been trained in such a way, they may call as LLM, large learning language models. So there they will be feeding the content. These are the contents will be fed. So once I throw some questions, they will be have written some programs where that based on that program, it will be giving the answers. So some of the what do we call AI, artificial intelligence, it shows intelligence right. If you go to Google and if you try to type BENGA, it may be Bengal also, right? But I'm saying in Bangalore or I'm in the map location of Bangalore. So what it says, Bangalore, what are the possible permission combination it will be giving. This is one type of AI. Next one is language translation. So I know only English. So if I'm coming from other state or some places, I just want to discuss with some auto driver or some place where I have to go. What I can do is I can go to Google and I can ask a question. So for good morning, Shubhodaya, it gives right. And moreover, you can able to see the audio also. It will give the way how to pronounce. Maybe here and there it will be, but it will try to give its best. Your face recognition. That is also another AI. Because all the things will be connected as a neural networks. Based on the network, if I keep then only our biometric systems, face recognition, right? Mobile, if you go, if you what do we call? Just if you show in a fraction of a second, it will open, right? That is another AI. So virtual assistants also can be called as, because we know about the some iPhone users, they may call us, hey Siri, call someone, play some song like that. So Amazon Alexa. And then, hey Google, I'm Cortana, that have died because it's in the Microsoft. Now they have updated to Copilot, such kind of things they have updated. So this is another type of AI. So email filtering, we know our emails, right? One will be main emails. Second one will be what I can able to see, primary, social, promotions, updates, such kind of things can be able to make it out. So that kind of auto filter, if some mail comes from a promotional point of view, some comes as a bank, it will go to update, some come directly from someone personal mail ID, then it will go to our primary mails. So this is another type of AI. So then image to text converter, if the font is there, right, based on the font, example, if you upload any document which is handwritten to an what do we call websites, what happens there is automatically the script will be converted. You can able to convert them. That is also another type of AI.
Game, a game of trial and error. Often scientists can only guess the mechanism that's causing an illness. From initial molecular discovery to bringing a new drug to market, it takes at least drug discovery.
So what is the timeline for AI, where it started? It's not a new term, and this was when the term was coined in the year of 1955. The father of AI is John McCarthy. So it's not a new one. It was available from the long period. So if you look into the history story, the first AI machine was used during World War II. The machine name is called the Bombay machine. And then next in 1950, they started to use by Alan Turing. And then Father McCarthy in the conference, they have made an American computer scientist have coined the term called as artificial intelligence during the first ever AI conference at Dartmouth conference in 1956. And later, the AI programming language was launched in the year of 1958. So this have been started. And first industrial robot named as Unimate was launched, and the first chatbot was Eliza, as the chatbot which was used. And the next one is the chatbot Alice, that was invented in the year of 1995. And then 1997, Man versus Machine, a Deep Blue beats chess legend. So the computer, they have trained the program to play a chess. So a person who was a world champion, he played against a computer and he lost the thing. So that means that there are such kind of things they have developed. So, and moreover, Kismet, this is emotionally equipped with a robot. The emotion things have been connected toward those things and all. Like you might have seen the movie called as Endiran, the Rajini movie with these things. They'll be feeding, right? The such kind of thing was happened during the year of 1998. And then in 2002, they started to use a robot for cleaning the floors. And then in the 2008, iPhone 3 and other things came. By hearing our voice, by recognition of our voice, it will be doing the works. So then Jeopardy in the 2011, it came. So which wins the quiz against the human champions. And then Amper Music, the first A music composer. How the music composer have been takes place. The same way music composer AI was launched in the year of 2017. And generative pre-trained transformer open AI, that is our chat GPT. GPT, you see, right? That is the full form is generative pre-trained transformer. That is the term. So it was launched in the year of 2018. And 2020 AI continues to advance rapidly, impacting various industries, including healthcare, finance, and automatic vehicles. So these are the various regions, various places the AI has started to evolve. So you know about the AI. The recently Tesla came, right? A man can simply sit and make a location, automatically go off. That is an AI. So where it is used, astronomy, it can be used in the field of bioinformatics, it can be used in the field of chemistry, it can be used in the field of evaluating the human resources, that is HR policies. Nowadays, the HRs are not sitting and recruiting or visualizing the resumes. What they will do, they will be uploading to the system, they will make a program based on your profile, it will be giving you a marks. So if the marks are passing, then only they will go for the next round. So 1,000 resumes may come, they can sit and do right. For that, they are utilizing the AI. And transportation, manufacturing process control, signal and image processing, speech recognition, and games, and more. So these are the various places that AI is playing a major role. So what is the AI application in the pharma sector or in the field of pharmaceutical industries? So first thing is where it can be utilized in the sense faster and better diagnosis. Olden days, if you go for some diagnosis like MRI scans or something else, the doctors will take some time to identify because certain diseases you can't predict at the accurate level. So what they started to do is they started to take all the images, they will be trained to the system. They will be feeding the system like this is the condition, this is the outline, how a person's brain will be, how the neural network will be present. In case if there is a block here, this kind of disease. In case if the block is over there, what type of disease, like that they will be trained. So once the model is trained very well, what I can do is I can able to take a report of a person, someone else, and I can upload into that, and I can ask a question, find out what happened. So is there any fault with this person, or what type of disease he is facing, what type of diagnosis has to be done. Like that, if you give right, it will be giving you the best output, like this person is suffering from so and so. So further in this field it can be utilized. And clinical trials process optimization, so much errors are there in the clinical trials. To optimize that, we can able to utilize the support of AI. Then in the drug development, which is playing a major role. And then in the field of patient monitoring and care, a dosing, and then the recognized depressions, precision medicine, risk assessment based on the symptoms. So this will recruitment for clinical trials, manufacturing process optimization, and business intelligence, etc. So these are the various places it will be working out as a support system. So where this will help in the sense, the peoples are doing a case studies and so many case studies they will be taking and they will be feeding into a particular system based on the available data. The system will try to generate some models like what type of mentality is, whether it's so much depressed due to this condition. So such kind of things can be predicted. And risk assessment, it can be done. And moreover, adverse reactions you can able to predict, you can able to pick the dose, and you can able to monitor the patient. You will be getting messages, right, reminder on time. Such kind of things can be done using the AI. So what is the advantages of AI? So it's more powerful and more useful than with the help of computers. New and improved interfaces will be available, solving new problems, better handling the information, release the informations of overloaded, and then conversion of information into knowledge. So we may have bundles and bundles of books. If you go to the hospitals or something else, medical records will be present. Where with that records we can able to make it out. That's our resources. That's an information. But if I take those data and if I feed into the system and I try to calculate a range, if within this range, what has to be done? If within this range, what can be done? So such kind of things can be predicted and can be able to evolve. So for such kind of data conversion or converting the information to knowledge, AI plays a major role. So in general, how it will be utilized in the drug discovery process? So the general drug discovery will be like this, our regular drug discovery process. So our general drug discovery process will be target identification, lead discovery, medicinal chemistry, in vitro studies, in vivo studies, clinical trials. This is a routine process where AI can able to support in the sense for the target identification. Yesterday, what we have discussed, right, proteomics, genomics, and recently for AlphaFold identifier, the person who got a Nobel Prize, because their deep learning he have utilized to model the protein. So such kind of proteomics, genomics can be done. Virtual screening, molecular docking, MD simulation, QSAR, preclinical simulation analysis, cell culture analysis, patient analysis, and statistical analysis. All kind of things can be utilized with the support of AI. So these are the various places AI can play a major role in the drug discovery. So now I'll be entering into the topic called as QSAR relationship. So what is the role of QSAR and those things? So I want to perform a study and I have designed some chemical structures of the same nucleus. To an example, I have a beta lactam ring, beta lactam compounds like penicillin, cephalosporins, and all. I have designed a compounds, but I don't want to go with a method called as molecular docking directly. So prior to that, I just want to check whether this molecule will be more effective or not. Underline the word more effective. In docking, it will be giving whether it will bind or not. More effective in the sense at what range it will be working out. So to predict over those things, and I'll be utilizing the technique called as QSAR modeling. SR is the first 1G, QSAR is the 2G second generation, and AI/ML is the third generation process. Where first SAR just by seeing the structure will be saying, but QSAR will be taking only the selected properties and we won't go with the multiple properties. But in machine learning, this will take all the best features and it will try to generate an equation to predict a unit synthesis compound at what nanomolar range or what micromolar range it will be acting. Such kind of things can be predicted. So here QSAR, quantitative structural activity relationship, this is the theoretical model. So as I said, right, I just want to work with a beta lactam ring and all. So how to perform? So I have planned to take a beta lactam and I just want to do an inhibition study for DNA gyrase. That is my target. So what I can do is I can take any published articles where the concerned researchers might be what do we call? They have synthesized the beta lactam ring containing compounds and they have carried out the in vitro studies with the same what do we call IC50 value. Let it be an example, or enzyme inhibition concentration. So that will be the thing. If the data set is 20, it won't be sufficient for me, because as how much we are increasing the data set, we'll be getting the better models. So for predicting those things and all, we can able to utilize those things. One article from one author, another article from some other places also you can take. But here the only one condition you should follow, the analysis of, that is in vitro analysis, the evaluation method should be similar. Example, for acetylcholinesterase enzyme inhibition method, there is a method called as Ellman's method and Ellman's et al. What we call modified Ellman's method. So these two are similar? No. Some modifications they did in the sense the results may vary. So that if you are going to choose a method in the sense, Ellman's method means same method compounds only you have to take. So from one article 20, another article 20, another article 20, 60 compounds I have got. Among the 60 compounds, what I have is some 15 may be potent, some 20 may be moderate, and others are poor in activity, or else no activity was found. So the same nucleus, and like bit. So all the nucleus are beta lactam, right? Beta lactam containing. But why there is a difference in their activity point of view? As a common nucleus is there, right? The skeleton is common. But even though it may have not shown the activity, why? We have to find out those reasons. That is called the descriptors, which are the descriptors playing a role. So for that, we'll be utilizing this method. So 15 potent, 20 moderate, and 35 are it's not working properly. So that's our outline. So what I will do is I'll take all the compound and I will be converting their IC50 value into PIC50, predicted IC50 I'll be converting. So that predicted IC50 based on the nanomolar range, I'll be converting as a log. Predicted IC50 is logarithm of whatever the value we are having, that will be converted. So that process will take place as a first one priority. So then what I'll do is I'll be converting these structures into smileys. Once the smileys are converted, so based on the smileys, I'll be generating the property. To generate the property, there are various tools available. Example, PaDEL is the tool which is freely available. And rather than that, if you go to a database called as ChEMDES, chemical descriptor, so there you can able to generate n number of properties, so that you can able to make a great amount of descriptors. So example, moderate if I'm using, I can able to generate 1800 descriptors. One chemical structure, I can able to generate 1800 properties. So that I can able to get it done. And then next one is PaDEL. PaDEL it will be working with the what we call Windows system also, and moreover it can be worked with the Linux system also. So this how I'll be generating the property, and based on that property, X versus Y versus X, such kind of concentration. That means Y is the value. This chemical compound showed this much activity, that is IC50 value or PIC50 value which is converted. But the structure is there, right? That structure is having properties, various properties. And yesterday I showed a cycle, topological property, electronic property, conformational property, such kind of things. So each and every property will have so many descriptors. So in that, which is playing a major role in producing the biological activity, that I'll be predicting. So once I have predicted those things and all, I'll be generating a model. With that model, and unnet synthesis compounds will be the right. What I have thought in my mind, what I have designed, that compound, I'll be predicting the properties, generating the properties. That data, if I go and substitute, it will be giving the value of at what range this drug or this chemical structure will be working. That's the outline. So these are the theoretical model that related to a quantitative measure of a chemical structure to a physical property or a biological property. So biological property is equal to F is equal to F of that is physiochemical properties. It's a collection of physiochemical properties. So this is what we call HAR and QSAR. What I said, as I said, right? So controls the potency, add the gram positive activity, control the garage bacterial activity. So each and every functional groups will be playing a major role in producing one type of activity, that is called as SAR. So in that, what is the distance between those things? If I calculate, that's another property that is called as QHER, molecular descriptor generation. And then how many number of donors, how many number of acceptors should be present, that will go as a QSAR model. So now I'll be entering into the term called as machine learning model. What is the difference between QSAR and SAR? Or also, what is the difference between QSAR and machine learning if you ask? So QSAR if you are running with the software, it will be trying to take only 2 to 3 major properties from the descriptors which we have generated. Example, as I said, right, 1800 descriptors we generate. This will consider only 2 to 3 properties. Why? Because it will take more time to generate, as well as it will take what do we call? It may not able to give the better output. The R square value should be the thing. If we are not going to get the better R square value, that model is not a best one. So always here we'll be generating the equation y is equal to mx plus c. That the equation you have studied, right? A linear graph should become. So the same way here also we'll be working. The R square value should be near to 1. That means 0.9, like that we have to get. So that will be the best fit or best model. So that's a QSAR. Now machine learning. What happens here is, in QSAR, I'll be taking one set of molecule. That means acid, what do we call, beta lactam compound content. But in machine learning model, I can take n number of compounds, n number of derivatives, n number of scaffolds, diversified scaffolds. Not only beta lactam, I can able to take any kind of ring structures. Pyrrole I can take, pyrazole I can take, acetone I can take. So n number of structures which already have worked forwards that particular target which I'm going to focus. So I can take diversified compound list, I can able to generate the property, and I can create an equation. The one new what do we call, only one nucleus. Here multiple nucleus I can take. Here very few properties will be generated or utilized to create an equation. But here multiple properties it will take. The best values it will be taking, and it will give us the best equation, best values. So machine learning is the field of study that makes the computers capable of automatically learning and improvising from the experiencer. So this focus on the strength of the computers with the help of collecting data from various observations, multiple observations which we have, right? Based on that, we can able to make it out. Machine learning is defined as a technology that is used to train the machines to perform various actions such as prediction, recommendation, estimations, etc. We can able to make it out. So here it will be thinking like a human. So what type of models are available? As I said, right, algorithm. Same way here also we have different algorithms, different models will be there. One is SVM model, another one is KNN model, random forest, tree model, AdaBoost, neural networking, such kind of, it goes on. So if you calculate them, it will be of a deep learning model or also supervised learning, unsupervised learning, such kind of things will be presented. So first one is neural networks. So as I given so many things, right, just I will give an outline. So neural network will simulate the way how the human brain works. We have neural networks, right? The same way it will work with a huge number of linked processing nodes. It will try to connect all the nodes and edges to get the best output. So it's a good at recognizing the patterns and play an important role in applications including natural language translation, image recognition, speech recognition, and image creation. For this thing, this will be utilized. And decision tree. So the word itself says tree, a decision. So what it will do? So decision trees can be used for both predicting numerical values, regression, and classifying the data into the categories. We may have the categories. So boy, girl. So which category it has to go, which category this has to come. Like in our Excel sheet, we may make a column, right? Filtering the process. The same way it will make a tree and it will give up. So it will be using a branching sequences. We know the generation, right? Father, he will be having son 1, son 2, son 1, one daughter, one like that. No, we'll be making a branches, right? This will always do with the branching things, and it will get us the best output. One advantage of the decision tree is that easy to validate, audit, unlike the black box in the neural network. So it is the simple method to get the best data output. And then the random forest, if you look in random forest, the machine learning algorithm predicts a value or the category, combining the results from the number of decision trees. The previous one it will utilize, and based on that it may give the advanced values. And then SVM model. SVM model, that is a support vector machine model. So what we'll do is, it's a robust classification and regression technique. Almost all are linear regression techniques or correlation. This thing only statistical methods only will be utilizing for any kind of tools. So here this regression technique maximize the predictive accuracy. More accuracy it will try to find out, and it will give the best results. So SVM is particularly suited for analyzing data with very large numbers. Example, thousands of predictors can be used. And then KNN, K nearest neighbors algorithm, K nearest neighbors algorithm, this will be utilized. Or it's a non-parametric supervised learning classifier. Previously I said, right, supervised and unsupervised. The same way here, this is supervised classifier where it will be helpful in grouping the individual data point. This data here, this data here, like that, it will be making out. AdaBoost. This is the Friedman developed a gradient boosting. This is to boost up the previous, what are the things we have predicted. So these are the various techniques which is available for doing a machine learning model. So how I can able to make a flow? What is the workflow I can able to do? So the workflow which I have a standard data set. So this standard data set, in the sense, now I have to decide which target I am going to work on, which protein I have to work on. So yesterday while discussing the neural network, network pharmacology, he said these are the targets I can able to work on, some targets for the disease, or the protein selection, he said, right. So I have an idea that I'm going to work with the acetylcholinesterase, because I'm working in that area. I will take that as an example and I will say. So acetylcholinesterase is an enzyme which is used majorly plays a role in Alzheimer's disease. That's the concept. So I just feel I have to work with those things, how I can able to work on. Point number one. Point number two is I'll go to a database called ChEMBL. That database will have the complete study where in any part of the world, any scientists, any researchers, if they have published the work on the paper for this particular structure, this is the value that will be available in the database. So from the database, I'll be downloading the complete history or complete data set. My data set will be very complete. So that I'll be making out. So I'll be showing the demo in a moment. So this is the ChEMBL database where I'll be giving my query. My query is acetylcholinesterase enzyme. My query is acetylcholinesterase enzyme. So once I give the cholinesterase enzyme, I can get whatever the possible compounds uploaded into the database, we can able to download. So once I have downloaded, my data sets will be like this: binding, ligand smiles, ligand its properties, such kind of so many properties will be presented. But here I need a very few data only from that. And moreover, that data will not be complete, or to be called a duplicate data will be present. I should pre-process the study. So that I should take place. So one is after selecting the targeted, I have to download them as a CSV file I can download. So once I have downloaded, that structure, that database will have duplicate values. Duplicate values will be present. Why? Because some may have tried with one compound, some may have tried with other methods also will be there. So that I should remove. Second, one researcher may focus to predict the IC50 value. Some researcher may focus to predict the EC50 value, effective concentration 50 value. Some may go for enzyme inhibition value, Km value, Vmax value, such kind of values will be there. So I have to focus only one output. So example, I'll be taking the IC50 value only. So that IC50 value only I have to work on. So this data once I get, I don't want the compound name. Only I need their smileys and their results, IC50 value. That two columns only I'll keep, rest and all I'll delete out. So once I have deleted all the things, I can able to convert these structures, because smileys I should convert as a structure, right? So I can convert them as a structure by using open Babel software. It's a freeware available where that tool is used to convert the structure from one property to another property. Example, file format will be there, mole file will be there, SDF file will be there, PDB file will be there, like how we are having PPT, PDF, Word, DOC. Same way here, these are the compounds which will be present. So this, what do we call, chem IDs will be the smiley compounds, all those things I'll be filtering. So now I can able to filter out. So this I'll be doing with our simple Excel only. There in a conditional formatting, if you click, automatically it will highlight. I'll show all those things. It will highlight which are the compounds are repeated. So many times it will be repeated, some assays will be presented. That I can able to delete easily through using Excel. After that process only, I'll be going to generate the smileys into structure. So I can able to, what do we call, I can able to make the compound into a single cluster I need. Single cluster I need. So that cluster making they'll be explaining during the docking. So in general, I'll be utilizing the tool called as BioWare Discovery Studio. which is a visualizer available free only. So that tool you can able to take and you can able to work on. So PaDEL. So PaDEL is a tool which will work on Java based. Every program will have run on one thing, right? Python programming. So such kind of things in the for a Word file or sorry, not a Word. For Windows, it will be utilized in a jar file only .exe directly you can run. I think some now it's not available in the databases and all. I have something. I'll share with the link. So if you go towards this thing, you can able to see so many options. You can able to see so many options: molecules, directory, descriptor, output file, 1D, 2D, 3D, fingerprints, remove source. So many properties are present over here where I can able to choose my molecules. I can upload them and I can able to generate the properties. To generate the property in our system, it will take some time. Why? Why? Because it depends upon the number of compounds which I have, and second one, how many properties I set to generate, either only 1D or 2D or 3D, what I have given. Based on that, it will take time and it will generate the properties. So PaDEL using what we call it can able to generate up to 1827 properties. Either it may be 1D, 2D, fingerprint analysis, whatever the analysis, it can able to generate up to 1827 properties. So once the property is generated, I have to filter, because as here I'm taking a data set, a multi data set, diversified data sets I took. So that data set will be generating the property, right? Where if example one property will be based on the nitrogen, one compound may have nitrogen, one compound may not have nitrogen. So for that nitrogen, the equation will have a value that will be generated for one compound. Another compound it may not be generated. Maybe that property is not suitable for that compound. So it will be 0, 0 or 0, 1 on and off like that. And so these things we have to sit and clear it up. As a manual work we have to do. We have to select that particular row or particular column and we have to delete all those things and all. After delete, what do we call? Sometimes that table will be empty. Table will be empty. So you can able to give a control H, empty space, put a 0. It will replace the empty spaces with a 0. So then based on that, we can able to select and delete the columns which we don't want. So once that is done, I may have from 1800 to some 600 or 800 table, what do we call properties. Now I'll be entering. This is the pre-process steps where I'll be utilizing first one is the ChEMBL database for collecting the data sets. Second, I'll be using Excel for filtering all the properties. When the smileys are ready, I'll be converting them into SDF file and there'll be or a small file, whatever the thing I want, I'll be converting using PaDEL. Once I have done those things and all, then I'll be uploading, I'll be filtering the unwanted properties, unwanted parameters, and I'll be deleting all those columns. So once that is done, next I'll be going towards the ML model. So Orange tool. This Orange is a data mining tool which is available for free to work on with any persons. It's a data mining tool. So this tool will have different platforms. Various models I said right. Tree model, gradient boosting, random forest, logistic, neural network, all stochastic regression, such kind of various models. Everything is a pre-built in this thing. Actually, if I want to say AIML, we have to work with the programming codes only. So it's like for a startup or to initial work, you can utilize this tool. This tool is working beautiful and it will give you a better results. But we have to keep on optimizing the methods. What will work out? How I can able to take the data? How I can able to delete the data? Making the data set only takes a large time. So here these are the available widgets. By using these widgets, we are going to we can able to carry out the ML models. So this will be the orange phase. Data will be there, data transformation will be there, visualization tool will be there, model will be there, and evaluation tool also will be present. Everything it is a pre-built simple steps. It is a thing, click, 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 like that it will go off. So example workflows, if you want, you can able to go to click and example workflow. It will be like that only. Just connecting the nets, connecting the dots. So once you connect, you can able to see various principal component analysis, hierarchical feature ranking, whatever the thing you want to do, you can able to take any models, any methods, and you can able to perform. So as I said right, almost some 600 to 700 properties will be present. But all the properties going to play a role? No. I have to filter all those properties. What I will do in the sense, which parameters played a role? Variations in the parameters much required. If it is of similar value, if I have a 10 compounds for one property, if all 10 are showing one means it's of no use. I have to filter it out. I need various diversified values. For that, I'll be utilizing the method called as a widget imputer. Impute is the tool which we'll be utilizing for filtering out the properties. So once you upload all your compounds, all your data set, this x1, 2, 3, 4, I said, right, they are the properties which you have generated. So based on that, it will, y is the numeric target. Y in the sense, our IC50 value, predicted IC50 value is the right. That value I should upload. Once I have upload and I have to change them as a role, that is a target. These are the features. Name may be meta. Meta in the sense, what are the, maybe I am kept as a compound 1, 2, 3, 4, 5. What are the thing I want to keep, I can able to keep. So based on this y value, all x will be correlated. It will go for a, what we call dimensionality reduction. Various dimensions will be present. Based on the dimensional reduction, it will give out the best. It will give out the best scoring. It will give which property played a major role, 1, 2, 3, 4, 5, like that it will be giving. So that particular property only I can choose. I can use that particular property for my machine learning models. So as I said, right, you can able to go with univariate regression or else or a relief method. So this you can able to make it out. So right, univariate regression, what is the thing, how far it can able to make it out, the difference value you can able to see. So that I can able to further, I can based on the ranking system, I can save them the top 50, top 10. I can give before only. So what do we call here? Best ranked, how many best ranked I need? 10, 15, 20, 100. What are the numbers I want? I can able to take it out and I can able to save to them. So this is the model, how a workflow, how it will be making, how a machine learning model will go off. So this file will be of our finally imputed file. I have imputed file, right? I have made a, what do we call, previous step. That file will be my inputs and selected columns will be there. So from here, I'll start to make a connected networks. I showed the widgets will be there. I'll be connected to gradient boosting, neural network. If I want to work with one model, I can work, but I'm not sure which model is going to be the best. So I'll make all the models to run. Just give a run and it will go as a test score. Scatter plot will be there, data table will be there. So this test score will give you what is the R square value for that particular model. Model 1, model 2, model 3, or maybe what do we call tree model, random forest model, what are the values you want, you can able to get those models. And based on that things. So once that is done, fine. My model I have validated. I have to validate till R square is nearby 1. So how I can able to do in the sense, I should check the data sets to which range of IC50 I have kept. So there I can able to delete or what do we call, reduce the data set or increase the data set. Based on that, if I come towards the R square value, so when that is done, I can save the data where I'll be getting the predicted IC50 value and the model IC50 value. Example, my compound, my predicted IC50 value is 1. My model was found to be better 0.9 in the sense I'll be getting the value as 0.9. Or if it is 5 means it will come as a 4.5 or 4.6, like that. That much range it will be giving. So once I'm satisfied with the model, I'm happy with the model. So you can able to validate, leave out one, leave out many. Such kind of options are there. I'll be showing through the demo. So once I have done all those data sets, this predictions will be the next option. Where here you can able to see a import file, right? This is my test compounds where I have generated the properties which I have uploaded. Same imputed properties I have generated for my designed compounds or the natural products or FDA products, whatever the thing may be. I may have the data sets and I'll be uploading without the y column will be empty, or else I can upload simply. So this I can able to use the widget called as predictions, the option. So I'll be connecting all the network because these are the trained network. This is the test. With this trained support, it will, once it is bound to this thing, it will produce the data table where the data table will be having your predicted IC50 value. So from that, I can able to conclude or come to an conclusion or idea that to which range, which nanomolar range or which micromolar range my compounds will work on. So those compounds I can take for further studies like molecular docking I can do. I can able to carry out MD simulation, and then I can go for in vitro and in vivo studies. This is how the workflow will be keep going on. So this is how my results will be coming. Mean square error, root mean square error, mean absolute error, and R square value. You can able to see the various models, and you can able to see the values. One model shows 0.31, 0.917. So from this I can say for my compound data set, tree model will be working better. So I can able to go with the tree model. And here you can able to do the cross validation studies. Number of folds, how many you want, 5-fold, 10-fold, 20-fold, as many folds you can want, you can able to make it out. And which method you are going to do, random sampling or test on trained data set, test on tested data set, leave out one. So these are the various things we can able to make it out. So PIC test for the test data set, how I will get output in the sense, my compound name I have my kept, and these are my properties which I have uploaded. And this will be the random forest value for the tree model, for SP model, and for me, the tree model is the best one. So I have taken the tree model and I'll be working on that. So based on this value, I'll be generating the scores, generating the data sets. What are the data sets are generated? That particular compound, I may have the smileys separately. That smileys alone I will take for further studies. So my results will be perfect once I carry out my docking studies, dynamic study, it will be fulfilled. Why? Because here I'll be getting the how much amount it will be effective. They are binding, how effectively it will bind, and the MD simulation, how far it will be more stable, so that my output will be far better. So this how I'll be working on the machine learning model. Huh? No. ಅದೇ ಬಟ್ ಇದು ಹಾಕ್ಬೇಕೈತಲ್ಲ ಅವನ್ ಎಲ್ಲೂ ಇದ್ದಾನೆ ಕೃಷ್ಣಗಂತೆ ಕರಿಯೋ Is there any queries still now? Any questions? Yes, yes. Yes, please.
You have, so you said that you are taking the data set from ChEMBL database.
ChEMBL database.
Yeah.
Ah,
So for test set, so for standard set, you are taking from a ChEMBL database.
I'll come over there. Little louder. No, I speak in the mic.
Sir, for standard data set, you are taking from ChEMBL database.
Ah.
For test set,
Test set, I can design any compounds. I can go to the Zinc database. I can go to FD approved databases, or what are the compounds available, you can take from any databases. So if you have a plan for any synthetic compounds, you can try. Or if you are working with the plant derivatives, you can take that as a tested data set. You can able to work on natural databases are available. That will be the thing.
oh, so while uploading in the PaDEL,
PaDEL.
yeah,
Ah.
and also in Orange, we want to upload both the sets.
Both in the sense?
Test set and as well as standard.
Yes, yes. Test set one from the what do we call obtained data set is there a standard data set. That standard data set will be helpful for creating an effective model. For the test, once you upload, it will based on this model, it will give the results. I have trained here. I have trained the model. So model is ready. Based on that model, now if I give this thing, it will give me the output that at this range it will be working on that. Yes. ಬಿಡು ಟೆನ್ಷನ್ ತಗೋಬೇಡ. ಎಲ್ಲಿ ಆಗ್ತೀಯಾ? ಏಯ್ ಬೇಡ ಬಿಡು. ಆಯ್ತು ಬರ್ತೀನಿ ಹೋಗ್ಬೇಕು. So this is the ChEMBL database. So this database, once you upload your data sets, or else you can able to upload your query setter. Battery. Hello. Once you upload the data, what do we call? You can able to make a query in this thing. ChEMBL is the right. Here, if I type acetylcholine esterase, if I start to type, it will be coming so many things. Board work ಆಗ್ತಲ್ಲ. Assume. ಬಿಡು ಟೆನ್ಷನ್ ಆಗ್ಬಿಡು. ಸೈಡ್ ಹೋಗ್ತೇವೆ. So here, if you're running, you can able to get so many names, so many contents will be coming. As the network is little slow, I couldn't able to make it out the data sets. ಎಲ್ಲಿ ಕನೆಕ್ಟ್ ಮಾಡ್ತೀಯಾ? Here, this database, they will be updating regular basis. So example, if a person from somewhere in some part of the world, if they have done the study and if they uploaded to, not uploaded, published a paper, any research article, means automatically they made a code to extract and keep into the data set. So this ChEMBL is provided by EBA, European Bioinformatics Institution. They are taking care of these things and all. So here you can able to go and search with any target. So you can able to see assays 1K will be there, means like so many options will be there where you can able to see various compounds, you can able to make it out. I don't know, some error is going on, so I'll update. Maybe they will be in the update process also. So if I give no, right, here this left side, you can able to see a term called as compounds. This compounds, you can able to see the compounds. These are the structures it's not showing. I think they're an updation process. So you can able to see the chemical structures which already they have uploaded, or some part of the world they might have done. So this data set I'll be exporting as a CSV file. If I what do we call, I should select all, and then I should able to what do we call, I can export. So it will take some time. When the results are ready, you can able to see the term. Click here and those things. So as it's taking a time process, I'll be going with what do we call, next step, which already we have made the pre-data sets. Once we collect the data set like this, it will be like as I said, right? So it may have the compound ID, the smileys will be present. So this is the chemical compound ID. And then next, if you look into this, what type of target they are choosing, because this data set which we took for PA3K alpha, or and then I can able to see this values, right? So till AC50, these all are the various value or based on the various methods. So these are the various values or various methods they might have adapted to do the assay. Maybe I'll be working to predict an IC50. I may be working to predict the AC50 value. What type of work I need, I'll be doing those things and all. So here I should choose a proper method. So most probably I'll be going with IC50 or value, or else KI value I'll be taking, and I'll be working on that. So then other than that, I don't want to know about the temperature, from where it is curated, what is the ID name, PubChem ID. So all kind of complete data will be present, and I don't want those things and all. Only IC50 value and smiley cyanide. Only those two I'll be taking, and I'll be filtering as a separate one. Now this will be my second data set. Step 2. From the previous raw data, I'll be taking only smileys and IC50 value. I'll be keeping ready. So this I have to convert into log IC50 value or predicted IC50 value. I can say why, because the range should be, if you see like a 0.07 and it will go on like anything. But if I convert them into natural logarithm or log of 9 minus log of this value, if I keep no, what I'll get is I'll be getting the various range. Why? Because if you take a log 10, what is the log 10 will be? 1, log 100 will be 2, log 1000 will be 3. This is the range. So 100 to 1000 will be 900 values. That all values will fall within only one range, one number, 2 to 3 only. So within the concise activity point of view, the researchers have started to carry out using the converted IC50 value. This IC50 value will be making out. So this IC50 value, I'll be just putting equal to 9 minus log of that value. That too, this based on the what do we call nanomolar range or micromolar range. It may be 3 minus log of, 6 minus log of, 9 minus log of. It varies, depends upon the compounds which we are having or the values which I have taken. As I said, right, if what are the compounds I have, the smileys, all the smileys I'll be uploading into the PaDEL descriptor just as a simple step. Once you upload, and if you select all the properties, what are the properties you want to generate, it will generate and give me like this Excel sheet. This kind of Excel sheet will be present. You can able to see the numbers. It will go like anything. It will be keep going on. As I said, right, 1800 plus properties have been generated. So So till it will be keep going on. So that much amount of what we call descriptors have been generated. So once that is done, I should filter the descriptors. As I said, right, even the 1800 descriptors are not going to play a role. I should reduce those descriptors so that I can able to get the best values. This I have done using the simple. Just go to columns, see whether the values are 0 or the similar value are present in means you can delete automatically that, and you can get this kind of data sheet. So till then, what I'll do, I'll be using the Excel sheet to generate all my data sheets. So next, I'll be going to use the orange tool to impute the property so that I can be able to get the best output or best properties I can able to generate. So this tool will take some time to start. Is there any queries? Is there any queries? So this is the orange tool where I'll be having all my properties and all those things and all. Here, as I said, right, my widgets. My widgets will be present data, transformation, visualization, model, evaluation, and unsupervised methods also. In this data, I can able to see what type of file I have to upload. Sometimes I can upload CSV, or sometimes I may have in a Word file, sometimes I may have in a TXT file, various file format will be there. So I can able to generally keep the word file and I can process it. So here I'll be what do we call? I'll be having a general file only I'm going to upload. So I have to so short right. So here you can able to see the numeric features. What are the thing you want to see? You can able to see what are the properties I have generated. That XR I just for my safer side. To I don't want to get confused with which property. So from 1 to last I give x1 2 3 4 5 like that, because y is my value and x will be my descriptors. So I made all those things and all, and it shows that. So you can able to see here. So if you want to change this role, because y always it should be a target. So once it is target and name is there, apply. So now if I impute all the values, or how to impute, either I need the average or most frequent values, or as the distinct value, as a fixed value if I give, automatically it will be imputed and it will be ranked over here. And it will be ranked over here as I showed in my previous thing. So I have given that best ranked 5. Down you can able to see the best ranked 5. Based on this, it have only highlighted the 5. So if I want whichever the other properties to be generated, or 5 to 50 or 500, 100, whatever the thing I want, I can able to take any numbers. Here you can able to see the properties. Almost 510 properties I have selected. From 1800, filtered and become 500. And now whatever the numbers I need, I can able to generate and I can able to make it out. So this value I can able to save as a data, save as .csv file I can able to save. So this will be the scatter plot which we need, because always we need the values right. It should be in the linear plot or scale. So based on that, this property it will be giving the values, and I can able to save this image also. And the only drawback with this orange tool is you can't able to export the images. You have to take screenshot, which poor quality it is showing. So this how I'll be imputing my data, so that from so many properties I'll be filtering only 50 or 100 properties for various molecules, and then I'll be going to use a method. Is there any questions? So this how I have generated my network. You can able to see the file where I have uploaded my file type and select columns, means which columns should be my X and which column should be my Y. Targets and features I should select, and I have given. So these are the various what to be called models: gradient boosting, neural network, random forest, whatever the thing may be. So that I can able to get from this model. So just go towards this model. Example, I want to run a random boost. In the sense, I can click, drag. That's it. It's a simple to connect all those things. So such kind of things I can able to make it out if I want. Then each and everything I can able to do. So from here, evaluator, test and score predictions are there, right? Here test and score and predictions. This test is running. That's why I couldn't able to show. So here this model should say me the value. What is the mean error value, absolute mean error value? What is my R square value? Based on that best model, I can able to satisfy. Okay, this is the thing. Then I can able to test and train data set as a for the cross validation. I can check whether the generated model is right or wrong, because if you go for a publication point of view, the reviewer will ask, have you done the validation? Validation, yes. Test on train data set, train and test data set, like that I have done, and this is the R square value. So once it is done, this model will be saved. And here the CSV import is the right. This CSV importer. This CSV import is the where I'll be uploading my test data set. My test data set, right? My test data set can be anything. Natural databases I can take and I can generate the properties using PaDEL or Moderate or ChEMBL, which are the tools I want, I can able to utilize. So once that is done, the next one I can able to go is I just simply put a compound name, their X values, that means the property values, and if I connect them, I have, you can able to see the connections. What it says in the sense, once this model is generated or developed, that it can be utilized for this data set. So if I collect and give right here, automatically it will predict and it will show me the big table where for radiant random forest what is the value, for gradient boost what is the value, for KNN what is the value. Such kind of data sets will be generated over here, and I can save them as a data table. So say save data if you give, it will be giving me output, where my output will be. So example, the test data set he has, I have generated from Zinc data, Zinc ID. Zinc, I went to Zinc database where I collected some natural data set, and I have made there only smileys. Only smileys I have. For this smileys, I have generated the property. So like this, I can have generated all the properties. And here why I kept as a y as an empty column, so that it will automatically predict my y value, and next to this columns will be always the answer that is predicted IC50 by the model I have developed using this orange tool. So that will give you an outline. From that I can able to keep an idea. From PIC50 I should again go back to this IC50 value. 9 minutes log of IC50 etc. Again reverse I have to do. So from that I'll come to know what is the nanomolar range this particular compound or this particular drug will, or what about synthetic or phytochemical can work on. So that will be the output. So from there I can able to save the models, I can able to move on. Example, I got some 2000 compounds I have uploaded in a database, or 2 lakh kind of I will keep a range from maximum 10 nanomolar or 15 nanomolar, 100 nanomolar range. I need the study means those compounds which showed 100 nanomolar range in the predicted IC50 value, that compound alone I can take as another data set. I can go for molecular docking and molecular dynamics and other studies and all I can able to carry out. This is how a ML work will be flowing. high level. Good, good. Bad. Is there any questions?
Good morning, one and all.
Yes.
One and all, myself, Anguraj, full-time research scholar from SRM University.
Little
Uh,
closer.
Yes, thank you so much for being the best, one of the computational chemist and teaching from basic to advanced level, sir. I have some queries. My first question is here, to develop the 2D QSAR, shall we take 3D molecular descriptor? No.
No, it is 2D or 3D or till if you look into the QSAR studies data, till 70 they are working on. 2D and 3D properties will be taking and working on. What is the difference between 2D and 3D in the sense I said right like this and like this, 2D and 3D. Why 3D plays a major role in the sense most probably our body is biological system said right, it will be in 3D nature. So 2D will show most of the physicochemical properties, but 3D point of view, all the angles, dihedral angle, torsion angle, that will play a role. So that is different part. This is different part. You can take 2D descriptor separately and work on 3D descriptor separately and work on 2D and 3D descriptor together. Also, you can work on. Here, the output is which properties plays a major role.
Okay, sir. Second query is, sometime I search the literature. In that one, sometimes that activity will be not accurately. Sometimes they'll give less than 1.
That we should not take. That we can't able to consider, right? If they said more than 10, means it may be 100,000 lakh also. That we should not consider. That we have to what to call remove.
Okay, sir. Next question is, in same literature, some diversity of activity. For example, one common have less activity, another common best activity. Shall we take all the range?
Yes, we have to take because we are going to generate a model. That means all past if it is present, there is no use. Plus minuses should be present and the average should be present. So always the thing is for a equal ratio is required. Potent, moderate potent, not active. That is required.
Okay, sir. Finally, I want your point of view, which is, one is a machine learning method, and another one is a like AI machine learning method. As per the validation property, MLR have more validation property like R square and concordance correlation coefficient, so and so. But machine learning, it has not that much. So, what is your point of view? Which one will be best?
ML will be goes best. Why? Because you, as I said right previously, that will go with only specific scaffolds. But here, this will be giving to a chemical structure what properties matter for producing this biological activity. So here in ML, we can able to get those answers. But in QSAR, we can't get those ideas. So the advanced techniques are there, right? That plays a role. So here it will be like a n number of components, and more or now what I showed is the initial stage of ML, and we have worked with so many things, and we have developed models with Python program. That will be too tough for people to understand only. We have that system is there, right? That is having so many of our content. Where if you work out a heat map, you can generate. We can able to make out so many models. PKL will be there. So once you start to work on, you will get more ideas. More data will be there. If you have more data, it will be better understanding, better output will be there. So always this will be better. And QSR, I'm not saying blaming on those things. It's a second generation. Now it's a third generation. That's the only thing.
Thank you so much for.
Thanks. Audience student.
My small concern with the software is that can we use molecules which we are designing for
Yes.
this also?
Yes.
It is
The
not-
test set I showed, right?
Yeah.
The test set is your design molecules.
Smileys we can put and we can.
Not with smileys. Smileys data you have to generate
Okay.
using PaDEL. PaDEL or else online tool can be open. So ChEMBL is the online server where you can, oh, they are also in the updation process. So ChEMBL, if you go, you can able to see the paper also. They have published a paper in Journal of Chemical Informatics. This tool is also having n number of properties. You can able to work on with those things and all. If you go upload your smile, but the thing is you have to wait. Patience is much required during this process. So if you upload into the data set and you can able to get all those data, you can able to make it out. From here, you will be getting your test properties that you can take them into the machine learning model and you can able to predict your compounds values, IC50 value or KM value, which your test sets your standard data set. Based on that, you can able to make it out.
So here we have one online question from Dr. Somya. Will regular configuration laptop supports all these tools, or do we need iConfigured?
No, this till this work or next sessions, next to 2 sessions will be with the ADMET and molecular docking. All this we can able to run with our own laptops. It is absolutely fine to work with the laptops or your if you have desktops also, it will be working with all the things, sir. But if you go for MD, then only we need the high-end software, sir. And moreover, your systems may be fast or slow. It all depends upon the GPU cores. Core size will be present, right? Based on the core size only, it can able to make it out of. So it is absolutely fine to work with your laptop. Thirty, 30, 11:30.
Any queries to be clarified? Any online questions?